Hello students, so welcome again. So we are dealing with the next chapter, that is the structure of DNA. So basically DNA, it is a deoxyribose nucleic acid which acts as a genetic material in most of the living organism except some RNA, that is a retroviruses. So the fragments of DNA is called as genes, which is considered as a single unit of heredity. So the sequence of a gene transcript into mRNA and then translated into protein. So the damage in the DNA can be repaired using DNA repair systems. So the DNA is a polymer of 4 different nucleotides, and the nucleotides also have some other functions, like we can say NAJ carriers, ATP, GTP, signal transductions, okay, coenzymes are there, various etc. there, okay. Now the next, it is made up of a nucleotide and the nucleosides one. Now what are basically, we will see it. A nucleoside is made up of a pentose sugar and nitrogen base, while a nucleotide is made up of pentose sugar, nitrogen base and a phosphate group. Okay, in the picture you can see. So the DNA consists of deoxyribonucleotides, while the RNA consists of ribonucleotides. Okay. So the first thing, that is the sugars. So the deoxyribose is a cyclic pentose, that is a 5 carbon sugar occurs in a DNA, while sugar in RNA is a we can say as a ribose one. So the next we can say the nitrogen bases. So in this, the nitrogen bases are the nitrogen containing aromatic heterocyclic compounds, so the purines and the pyrimidines ones. So these are the 2 different types of nitrogen bases found in DNA and the RNA. So adenine A and the guanines G are purines ones, while the cytosine, thymines T these and the uracil also U are the pyrimidines one. So in this we can say the thymine is replaced by uracil only in the RNA only. So next, that is a charge of rule, we can say. So this rule states that the DNA from any cell of any organisms has a ratio, that is a 1:1 of pyrimidines and the purine bases, and more specifically that the amount of guanines purines base is equal to cytosine pyrimidine base, and the amount of adenine, that is a purine base, is equal to the thymine, that is a pyrimidine base. So we can say, so the base pair is composed of a pyrimidine base and a purine base. So this pattern is found in both strands of DNA and is responsible for the base pairing rules. So which states that adenine always pairs with the thymine and guanine always we can say pairs with the cytosines. So the amount of we can say, so the total amount of adenine released is equal to the total amount of thymine, and we can say similarly the total amount of cytosine is equal to the total amount of guanine. That is, we can say A is equal to T and C is equal to G. Okay, these are the equal one. Now next, that is the C value paradox. So the C value is the amount of DNA in the haploid genome of an organism. So it we can say it varies over a very wide range with a general increase in the C value. So with the complexity of organisms from prokaryotes to the we can say to the invertebrates or the vertebrates or the plants, the C value paradox is the paradox in the amount of DNA and complexity of organism. Okay, while in the eukaryotic genome size fails to correlate with the complexity organism. So the largest genome we can say is found in amoeba, a single cell organisms, as well as in the human genomes, and one found in the yeast one. Okay, so this is the C value paradox one. Now Watson and the Crick model of DNA. So these are the scientists, we can say. In 1953, American biologist, that is a J.D. Watson, and the British physicist, that is a Francis Crick, proposed the 3-dimensional proposed the three-dimensional model of physiological DNA, that is it also known as a bDNA. Okay, so on the basis of X-ray diffraction data of DNA obtained by the Franklin and Wilkins, so on their basis they have done this. So for this we can say approach discovery, Watson, Crick and the Wilkins got the Nobel Prize in the medicine in 1962. So the terms DNA given by them only, by the Zachary's. Okay, so the important features of Watson and Crick model on a double helix model of a DNA RS, we can say some of the characters it is there. So the DNA molecule consists of two polynucleotide chains, or we can say strands that spirally twisted towards around each other and coiled around a common axis to form a right-handed double helix. Okay, in the image you can see. So the two strands are anti-parallel, that is they run in opposite direction, so that the three end and the one chain facing the five end of the other direction. Okay, where the sugar phosphate backbone remains on the outside, while the core of the helix contains the purines and the pyrimidine bases. So the two strands are held together by hydrogen bonds between the purines and the pyrimidine bases on the opposite strands, we can say. So the base composition of DNA obey in this condition we can say it obeys the Charles rules. Okay, according to which the A pairs with the T by two hydrogen bonds and G pairs with the C with the three hydrogen bonds. Now this the diameter, when we say in the picture you can see the diameter of the DNA is 20 nm, that is a 20 angstrom, and adjacent bases are separated by we can say 0.34 nm, that is we can say 3.4 angstrom along the axis. Okay, so the length of the complete turn of the helix is 3.4, or we can say 34 angstrom. So there are 10 base pairs per turn. Okay, so the DNA helix has a shallow groove called as a minor groove and a deep groove called as a major groove across the length. So the characteristics of the DNA, there are very important characteristics. The first, that is the amount of DNA per nucleus is constant in all the somatic cells of a given species. Okay, so the total amount of DNA is we can say in a haploid genome is the characteristics of each organism, which is known as a C value. Only a small fractions of DNA is functional in eukaryotes. So the DNA in the chemical basis of heredity and is organized into genes, or we can say cristones also. So the DNA replicates to form DNA and transcripts from RNAs. Now another character, that is a DNA replication occurs in the S phase of a cell cycles. Okay, then the DNA replication is a semi-conservative in which two daughter DNA molecules form. So the each receives one of the we can say parental strands and the another, that is a new strands one. So on one strand of DNA there is the synthesis of called as a template strands, or we can say antisense or non-coding strands. So the other strands is called as a coding or non-template or sense strands, which has the same sequence of the RNA transcripts except T in we can say in place of U. So that next, that is a DNA has many repeated base sequences commonly called as a satellite DNA. Okay, then DNA can easily undergo denaturations, or we can say melting and renaturations with any change in pH, temperature and salt concentration. So the DNA in with the high we can say G plus C, that is the RNA plus cytosine contains is more resistance to the thermal melting than the adenine thymine rich molecules. Okay, so the this DNA can be synthesized in vitro, that is in the laboratory ones, and last that is we can say it DNA can be measured by the unit picograms. Okay, so these are some characteristics of DNA. Now it has a different types of DNA presence. So after the discovery of DNA double helix in 1953, so this DNA was thought to have a typical B forms. So the Watson Crick model everywhere in all conditions, but later on, after some few 20 years, it was found that there are various experiments led to the discoveries of different forms of DNA were found out. So there are different types of DNA, that is A form, B form, C, D and the Z. The major one, that is the A, B and the Z one. The first, that is the B form DNA. So it is a biologically important form of DNA which is naturally found in most living organisms. So this form of DNA can be found in other different forms such as A and Z forms, which is conditions like relative humidity, ionic strengths, salt concentration and sequence of nucleotides. And other forms differ from B forms in features such as number of base pairs per turn, distance between the two base pairs and the diameters one. Okay, so the next, that is helical. If you see the difference between the helical side, so it is a B, it is a right-handed, A is right-handed, whereas the Z, it is a right-handed one. Okay, but if you see the conditions are in them, so 92% relative humidity, low ionic strength is found in the B form, whereas 75% relative humidity and the presence of nitrogen, potassium and ions are found in the A form, whereas very high ionic strengths are found in the Z form. Okay, so helical strength, since we have discussed now only. So base pair per ton it has a 10 base pair per ton, that is in B form, whereas in the A form 11 and in the Z form 12. So vertically raised, we can say base pairs is 3.4 Armstrong in the B form, then 2.56 Armstrong in the A form, whereas 3.71 Armstrong in the Z form. If you think about the rotation per base pairs, it is about we can say in extra conditions rotation, that is 36 degree plus or in the B form, whereas plus 33 degree in the A form, whereas we can say minus in Z form, it is shown that is a minus 30 degree. Okay, helix diameter, so it is in the B form, it is a 20, then 23 Armstrong in A, whereas 18 Armstrong in the Z form. So these are the types of DNA. In the picture you can see this. Thank you very much.